Welcome back, folks. Second part of the roles of ER and Golgi trafficking, and we're going to look at a specific example of trafficking here, trafficking of soluble lysosomal proteins to the lysosome, and this happens in the Golgi apparatus. And is under the control of two main enzymes in the Golgi. We'll take a look at those in a moment. And so the soluble lysosomal proteins that you find in the ER and the early Golgi compartments undergo N-glycosylation, followed by the removal of some of the glucose and mannose units in the glycosylation. Then some of the mannose sugars which are left on the soluble lysosomal proteins can be phosphorylated to produce mannose 6-phosphate, and that's the tag which ensures the correct delivery of soluble lysosomal proteins to the lysosome. So when I say soluble lysosomal proteins, what I mean is these proteins aren't transmembrane. So these are primarily acid hydrolases. Those are enzymes which break down proteins and other macromolecules at acidic pHs. So let's look at this process, and it looks a little bit complicated, but we can break it into some steps and make it much more manageable. So the phosphorylation of mannose to produce mannose 6-phosphate, which is the targeting signal to get these soluble proteins to the lysosome, is catalyzed by two Golgi specific enzymes. So these are enzymes which have to be kept in the Golgi, and so they're kept there using the mechanisms that we described before. The first one is a phosphotransferase that adds N-acetylglucosamine, which has been phosphorylated on its one carbon. So that's called N-acetylglucosamine 1-phosphate. And so that is added over here in the cis-Golgi network. So that's the first enzyme. So that's the addition of phospho-N-acetylglucosamine. And then the second enzyme works in the medial cisternal stacks, and that enzyme in the medial cisternal stacks is going to remove the N-acetylglucosamine part of N-acetylglucosamine, and that leaves 6 phosphorylated mannose. So mannose 6-phosphate. That's mannose with a phosphate group on the 6 carbon, and that's the signal, remember, that will take this specific soluble lysosomal protein to the lysosome. So in the trans-Golgi network, there are a series of receptor proteins. So these are transmembrane receptors, kind of like the receptors you find on the cell surface which bind a ligand, except here the ligand is the mannose 6-phosphate that's found on the surface of these soluble, soon-to-be lysosomal proteins. And so the mannose 6-phosphate on this protein binds to this receptor, and then a coat of proteins called clathrin produces a vesicle that contains the mannose 6-phosphate attached to the receptor protein. And so this little vesicle here is produced by this clathrin coat. And we'll talk about clathrin coats when we talk about coated vesicles and exocytosis and endocytosis a little bit later. And so the vesicle with the mannose 6-phosphate on the lysosomal protein attached to the receptor then uncoats, and this vesicle gets sent to what's called an early endosome, and then early endosomes develop into late endosomes through a series of enzyme maturation processes, and then they can fuse with mature lysosomes. So in this early endosome here, the pH is low, and so that low pH, that acidic pH, is generated by protons being pumped into the lumen of the vesicle, and so there's an ATP dependent proton pump here, and so that acidifies the lumen of the early endosome, and that causes the mannose 6-phosphorylated protein to dissociate from the mannose 6-phosphate receptor. So here's the receptor, and here's the mannose 6-phosphate containing protein dissociating. Then the phosphate is removed from the mannose 6, and you'll then end up with a lysosomal hydrolase, which will eventually be mature and fully active when it finds its way into a lysosome. So how then do we get these receptors back to the trans-Golgi network so we can keep this flux of these soluble lysosomal precursors being sent to the early endosomes, which can then become mature lysosomes? So the receptors get packaged into little vesicles which are covered by these retromer coats, and this targets the receptors back to the trans-Golgi network. So that's a very specific mechanism for moving a soluble lysosomal protein from the ER through the Golgi, through the trans-Golgi network, into early endosomes, until eventually that protein will find its way into a lysosome, and then we send the receptors back through this retrograde pathway. So this is anterograde, and this is retrograde, going back to the trans-Golgi network. So that's a nice example of a specific mechanism for sending certain soluble ER proteins to the correct cellular compartment. This doesn't happen on other proteins, only proteins that need to go to the lysosome. So if we're looking at a protein that's going to go to the plasma membrane, mannose-6-phosphate receptor, nothing to do with that. Okay, let's wrap it up there, and I'll be back soon with some more, and we'll talk about exocytosis and endocytosis.